for kind words. All dignitaries present over here, IWSA trustees, President Dr. Rita Mukhopadhyay, Lalita Dhareshwar Madam, and SIS College faculty members, authorities, and dear friends. The first slide which I have put here, this is the building where we have initiated founded our IWSA branch in Department of Botany, Shivaji University, Kolhapur. So this is the building, and topic of today's discussion is eco-restoration of mangroves. First of all, I would like to thank the organizers for giving me this opportunity and share my experience in the field of mangroves. Eco-restoration of mangroves is a wide aspect. It is academic aspect, and I will introduce about eco-restoration, the definition, regeneration techniques involved in eco-restoration, and some of the case studies. So you all know these habitats are unique strategic locations along the coast of Maharashtra. Because we have worked along the coast of Maharashtra, so emphasis is given along the coast of Maharashtra. Typical species are 27 along the coast of Maharashtra, and it has been divided into 6 coastal districts. Of course, there is still poor understanding of these systems, and you know there are numerous ecological values of mangrove ecosystems. They lie in protecting the coastlines from cyclones, floods, surges, sea level rise, wave actions, and coastal erosion. So they trap for sediments, sink for nutrients, and also they store and accumulate large amount of carbon. So these ecosystems also support the adjacent flora and fauna like corals, seagrasses, salt marshes, seaweeds, and other ecosystems too. All these ecosystems, they provide numerous benefits, and they have the source of food, breeding grounds, and nursery for fishes, shellfishes, and support number of organisms. And they remove carbon dioxide from atmosphere. And I'll give you an example, 22 metric tons per acre is accumulated through the process of photosynthesis, and it can be minimized the problem of greenhouse gases and our global warming. So nowadays, these ecosystems are degrading very, very, very fast along the metropolitan city. And therefore, eco-restoration of degraded areas along the coast of Mumbai, Thane, Raigad is very important aspect to be tackled. Eco-restoration is very important process of repairing the natural habitats and where the biological communities and species have been destroyed. Though it is an ecological aspect, we need to practice it very carefully. It's very difficult to practice eco-restoration programs along the coastal lines. It aims mainly to repair, revive, and accelerate the recovery of degraded sites along the coast. Now I'm going to highlight just few slides related to techniques which we have to practice for eco-restoration. That means eco-restoration is improving its functionality ecologically. That means we have to raise the reproductive capacity of that habitat. There are 2 ways. There will be natural regeneration and natural processes of establishments of seeds and propagules, and these methods are always advantageous and less disturbing. However, we can carry out artificial regeneration, but with formation of nurseries, establishing the nurseries at various sites along the coast, outside the coast, planting the seeds and suitable areas, direct and indirect plantation techniques can be carried out. And this technique is labor-intensive method. These are some of the characteristic features. There is a big family, Rhizophoriaceae, and the key character of Rhizophoriaceae is vivipary. Myrcinaceae, it shows cryptovivipary, and Avicenniaceae also shows cryptovivipary. These are some modes of regeneration of mangrove, as well as the key adaptations of Rhizophoriaceae, Myrcinaceae, and Avicenniaceae. Flower develops into fruit. There is a single seed, and to develop the seed, the requirement is for 10 to 12 months. It lacks resting period. Germination is always physiologically affect the higher concentration of chloride ions in the tissue. Well, these are the reproductive strategies. And as I have said, vivipary, it means embryo develops continuously while the fruit is attached to the mother plant. They may grow in place attached to the parent tree to at least for 1 year, that is 12 to 14 months, and reaching the length of 70 centimeters before detaching from the parent. And then as soon as there is a decrease in salinity during rainy season, they fall into the water and they germinate. Here are 3 species which are having the Cereopstragal, Rhizophora mucronata, and Bruguera gymnorrhiza, where they are having viviparous mechanism. These Avicennia and Aegiceras, they are having cryptoviviparous mechanism. It is hidden, but when there is moisture content, then the leaves, the fruit wall will be detached, and you will see the plumule are extending out. So modes of reproduction of vivipary, cryptovivipary, seed germination on soil, and vegetative propagation. So these are the saplings which we call propagules as a seed material or unit of propagation, which you use for ecorestoration programs. The above one is Cereopstragal, where you can see a very fine yellowish zone. This is called as maturation zone, and this zone, if it appears on the propagule, then we can identify that the propagule is ready for plantation. Likewise, there are small propagules of Rhizophora apiculata, and which we have shown it here. They are a little bit smaller than Rhizophora apiculata. This is the sapling material or propagule material which we are using here. The flower, fruit, single seed germination, then the growth continues. There will be development of radicle. That is at the point you will get the root developments. Also the fruit wall simultaneously develop, and along with that, there will be development of hypocotyle region, and then you will find this pod-like structure, which is unit of propagation. It is called as propagule. These microhabitats are very important because we have microclimatic regions in the mangrove habitat as they induce by environment various variations, which we call gene frequencies. These are morphometric variations, and morphometric variations are important as a biodiversity component because we have to conserve it as a phenotypic plasticity and which result of evolutionary process. It is a possible phenotypes. There is a wide range of variation, and they are more adapted and suited to that particular environment. So plus populations with these variants may be selected and identified as a perinating material. The size of the propagule varies as per species. The second technique is mangrove nursery establishment and management, which is involved in ecorestoration. To restore the degraded mangrove lands, direct plantation and indirect plantation techniques can be used. Designing mangrove nursery, establishment and operation of the mangrove nursery, use of stock for mangrove restoration, afforestation, and of course, we have to involve local participants or community Otherwise, it is impossible to carry out the restoration program. This is the proposed mangrove nursery plan, where you can see there is a walkboard and the plates have been made, and the side figure shows the soil and the propagules, how to keep these propagules along with the soil. This is not from India, this is from Malaysia. A huge voluminous programs have been carried out because Rhizophora is a successful species for plantation. This is from our area. Now they are tagging the well-developed established seedlings for plantation, and you can see the preparation for eco-restoration program. It is very significant aspect, that is selection of site for afforestation. Selection of suitable area, maps and drawings are necessary for every site, because each site is unique and different as per salinity regime. Knowledge of climatology of the site is very essential. Knowledge of phenology, that is art of observing reproductive phases, flowering phases, and all is very necessary for the eco-restoration program. Knowledge of natural zonation and knowledge of also species succession, because there are distinct zones in the mangrove habitat. This is just to highlight you about the sandy clay, rocky sandy clay, sealed clay, and which are the species they are growing in that particular area as a successional stages that I have shown in this figure, along with the salinity. This is the distribution and the growth pattern of mangrove seedlings along the shore, which can be easily make out as a successional phases of different mangrove species. Now we can go for the third aspect, that is the plantation technique. Plantation can be done by various methods. One is transplantation. We can establish the nursery in situ, ex situ, as well as we can go for direct plantation and indirect plantation also. Then trenching method is widely utilized along the south areas, south estuarine areas, because there are deltic mangroves. Dibbling method is also used, and enrichment of plantation by both direct and indirect methods can be achieved. This is one of the examples from Tamil Nadu, where they have formed the trenches and the plantation is carried out. Interior expanse of the plantation program along with nursery raised mangrove seedling. So this particular figure, it indicates various zones as per the succession. And now we have the Rhizophora plantation as a green belt along the coastal line. These are called as coastal sentinels or coastal guards, because they protect our shoreline. So they protect our shoreline as a guard, and hence they are called as sentinels of the coast. So this is how we can make green walls instead of constructing engineering walls, and then we can balance our ecosystem. Through this funding, we have started establishing the field gene banks for rare, endangered, and threatened species, mangrove species in Sindhudurg district. This is just to show you the germplasm conservation. Each and every species adaptation is different, key features are different, and these are the specifications. In June, July, August, you have to collect the
Dr.
fruits
Niranjana,
of Cyanometra iripa, and these are the collected germplasm of orthodox species, the other species, and the storage of Cyanometra iripa, collection and reservation of other species. And yes, madam. Yes. So just I will highlight you our work. The work is very vast, and species wise, we are dealing with the restoration programs. So this is how in situ and ex situ conservation have been attempted. These are some of the analytical part, control and the test plants. Jamdul Nursery at Achra. Again, this is for Bruguera cylindrica, which is also becoming rare and endangered. These are some of the attempts along the coast. Then about Rhizophora and Bruguera, this which have built up in Shivaji University, Kolhapur. There are various sectors as per the species, and successfully we have grown the nurseries. And I'm going to just highlight because lack of time. Bruguera cylindrica, Calophyllum, Elophyllum, Bruguera gymnorrhiza, Cereops, Dolichondron, Spathacea, Heritida, Littoralis, which is Sundri. This is a great achievement of our department. We have reported for the first time, and you can see here the eco-restoration program that we have dug it, the transporting of the raised nursery plants, then nursery raised at Jamdul and other areas. And now you can see here beautifully establishing surviving plants. And these are the workshops and the awareness programs from various themes, nursery techniques, field orientation program for forest officials and for students, and at Vengurle, at Achra, Malvan, Devgad, etc. So just we can go through the slides.
I would suggest that maybe you can show them when the discussion is going on at the end. Yeah.
So this is the site of Sundari.
Yeah.
And this is Vengurla site. We have done the plantation. This is one model for mangrove conservation and restoration. And at the end, there are various threads, as you all know, because you are staying in Mumbai. So how to save the mangroves? We have to save the mangroves, protect the mangroves, and balance our ecosystem, one earth and one being.